Ever wondered why the transforming growth factor beta or TGF-B pathway is so significant in cellular processes? This complex signaling network plays a critical role in regulating a variety of cellular functions, from cell growth and differentiation to apoptosis and immune responses. Its importance isn't just limited to normal development and homeostasis either. When this pathway isn't functioning properly, it can lead to various diseases like cancer, fibrosis, and autoimmune disorders. At the heart of the TGF-B pathway are the members of the TGF-B family of cytokines, which include TGF, B1, TGF, B2, and TGF, B3. These cytokines are secreted by various cell types and work as autocrine and paracrine signals. TGF, B1 in particular, is often referred to as the prototypical member of this family, renowned for its multifunctional roles in development and tissue homeostasis. These cytokines are synthesized as latent precursors, which need to be activated to become biologically active. This activation can be achieved through various mechanisms, including proteolytic cleavage or interaction with other molecules, such as integrins. The cellular response to TGF-B signals is mediated through a family of cell surface receptors known as the TGF-B receptors. These receptors are serine threonine kinases and include two classes, type 1 and type 2 receptors. The type 2 receptors, such as TGF-br2, bind TGF-B ligands, while the type 1 receptors, like TGF-br1, join the complex upon ligand binding. This binding induces the formation of a heteromeric receptor complex, enabling the transduction of TGF-B signals into the cell. The TGF-B signaling pathway can be divided into two distinct branches: the canonical or SMAD-dependent pathway and the non-canonical or SMAD-independent pathway. The canonical pathway is the most well-studied and involves a cascade of phosphorylation events that ultimately result in the activation of transcription factors known as SMADs. Upon ligand binding to the receptor complex, type 2 receptors phosphorylate and activate type 1 receptors. Activated type 1 receptors then phosphorylate receptor-regulated SMADs, specifically SMAD2 and SMAD3. Once phosphorylated, these SMADs form complexes with a common partner, SMAD4, and translocate to the nucleus, where they regulate gene expression by binding to specific DNA sequences and recruiting transcriptional co-activators or co-repressors. The non-canonical pathway, on the other hand, involves various intracellular signaling cascades that do not directly rely on SMAD proteins. Instead, these pathways are often characterized by the activation of other kinases, such as the mitogen-activated protein kinase pathway, phosphinosytide 3-kinase pathway, and Rho-like GT paces. These pathways interact with the TGF-B pathway to modulate specific cellular responses, including cell motility and apoptosis. To sum up, the TGF-B pathway is a complex and critical signaling network that plays a key role in various cellular processes. It is initiated by the TGF-B family of cytokines mediated by TGF-B receptors and involves both canonical and non-canonical signaling pathways. And while this pathway is crucial for normal development and homeostasis, its dysregulation can lead to a variety of diseases, underlining its importance in biology and medicine.